Hi, my name's Eli Weinstein, and today I'll be telling you about a project on optimal design of stochastic DNA synthesis protocols based on generative probabilistic models with my collaborators. So the setting of this project is that we're going to be thinking about generative sequence models, models that are probability distributions over biological sequences like proteins, and from which you can sample new sequences. And so our goal is to make as many samples from a generative sequence model in the laboratory as possible. So we want to make as many sequences physically as we can that constitute samples from some target sequence distribution P. So why might you want to do this? Well, one reason is, for instance, that you're doing protein or other kinds of sequence design. So for instance, you might be combining natural sequence data with some experimental measurements to produce a distribution over sequences predicted to have the property that you want, and then you want to synthesize as many as possible so that you can screen and check that they are indeed functional. As another example, you might be studying, for instance, pathogen forecasting. You've built some time series model of sequences, and then you want to test in the lab predicted future sequences, for instance, to evaluate the efficacy of some drug on future strains. In both these cases, we want to be able to make as many sequences as possible in the laboratory from our target distribution P, whatever it may be. So existing approaches to this problem, which we call Monte Carlo synthesis, consist of first drawing samples in silico, and then synthesizing those samples, those sequences, individually. And the issue with this approach is that currently for individual gene length synthesis, it's pretty expensive to go above, say, 10 to the 4th sequences, which is much smaller than many modern high throughput screens can handle, which could go to 10 to the 6th or 10 to the 9th or even above. So an alternative to individual synthesis is to use stochastic synthesis protocols. This would be something like error-prone PCR or mixed nucleotides or mixed trimer-based synthesis. These approaches can produce a very, very large number of unique sequences, and in effect only limited by screening capacity. So you can reach the full capacity of a screen. The issue, the challenge, is that they're random, these sequences. So our proposed method, which we call variational synthesis, is to write down a model of the stochastic synthesis protocol in terms of experimental parameters, so a distribution that describes the sequences that they produce, then minimize a divergence between that synthesis model and the target model P, and then finally run the stochastic synthesis protocol with the optimized parameters in the lab, and that gives you a very large number of samples from Q theta. And the key trade-off behind this idea is that instead of making a small number of exact samples, as in existing Monte Carlo synthesis, we're going to make a very large number of approximate samples. So in the preprint, we work through this recipe. We start by developing synthesis models for a number of popular stochastic synthesis technologies, including error-prone PCR or mixed nucleotides or trimer mixtures. We also consider techniques like combinatorial assembly. Once we have a synthesis model, Q theta, in terms of experimentally controllable parameters, we optimize that synthesis model, and so we developed a black box optimization procedure that works, or that can be applied to any target P, so long as you can draw samples from it. And it will optimize the parameters of these synthesis procedures. We developed some theory to quantify the trade-offs between variational and Monte Carlo synthesis. In particular, we showed under certain conditions, variational synthesis can lead to very, very large increases in the number of hits that you'll get in some downstream screen. In order to evaluate our variational synthesis procedure using detailed simulations, we relied on high-quality sequence-to-function predictors trained on large data sets. And based on these predictors, variational synthesis can lead to many more hits than Monte Carlo synthesis in practice for reasonable library sizes. So, for example, in this case, if you have a library set, if you can screen 10 to the 6th sequences but only synthesize 10 to the 3rd, variational synthesis will provide a 360x increase in the number of hits of fluorescent protein, and an even larger gain in this T-cell receptor screening assay. So, in conclusion, we proposed variational synthesis, which offers a strategy for producing large numbers of approximate samples in the lab. And if you want to learn more, you can check out our preprint here or our code there. And thanks for watching.